Okay, students, this is the last lecture of chapter 3, and then we'll have a quiz over chapter 3 next week, probably on Tuesday. You'll have a day or so to finish it. Okay? So if you recall, chapter 3, we're learning about LH and FSH and some of the characteristics of these two gonadotropins, and don't forget, we also talked a little bit about GnRH's mechanism of action. That was the first slide in lecture 3 of chapter 3. So if you recall, the main points of gonadotropins is there's an alpha and a beta subunit, and these two proteins are held together by non-covalent bonds. And the specificity of each hormone lies in the beta subunit, whereas the alphas are pretty generic, and the alphas could be switched back and forth between LH, FSH, and TSH without any effect. And so the specificity is within the beta subunit. And last time I left you, we saw this big behemoth receptor. This was porcine LH receptor. And so this receptor would be in the testes and the Leydig cells. This would be in theca cells in follicles. And this also would be in granulosa cells inside female follicles to bind LH. And gonadotropin receptors are members of the 7 transmembrane domain receptor family, if you remember that. So these huge receptors have a huge extracellular domain and a smaller intracellular domain. But remember, this is 673 amino acids long. It's pretty long. And so the hormone binds to this out here. And remember, we talked about there being 11 exons. So here's exon 1, 2, 3, 4, 5, 6, all the way to 11. And so let's count 1, 2, 3, 4, 5, 6, 7. So this membrane crosses this receptor, crosses the cell membrane 7 times, and that's why they call it a 7 transmembrane domain receptor. So for the most part, LH and FSH stimulate their target cells to produce steroids, testosterone, estrogen, and progesterone. However, gonadotropins also stimulate large numbers of other proteins key to specific functions of target cells. And so don't get to thinking that LH and FSH only cause steroids to be synthesized. For example, in females and in males, one of these proteins is another hormone called inhibin. And so that's an example of LH and FSH stimulating protein synthesis by their target cells. And so these proteins can stimulate or inhibit many actions and function of these target cells. So they're important as well. So what modulates or controls the target cells? So let's talk about modulation or desensitization of target cells. So as with many hormones, it's been shown that if you give an animal, or if an animal receives very high levels of LH, and in the case of this, they also use HCG. So in other words, another gonadotropin produced by pregnant females. So let's just stick with LH for a minute. So you give very high LH levels, we talked about this before, that will cause downregulation of the target cell and the target tissue. Well, what is downregulation? It's a very slow internalization of receptors. So the receptor we just saw, this target cell has the ability to pull that sucker back inside the cell and take it out of the cell membrane. That's not a primary role of any gonadotropin in nature, but in case the pituitary released too much LH, just in case it got too much GnRH and released too much LH, well, the target cell's not going to go crazy, it's going to become desensitized. And so most of these target cells are geared up to operate on very low levels of hormone. So if we give very high levels of hormone in an experimental model, what we see is a very slow internalization of receptors, and that's called downregulation. And so that would modulate the response of the target cell to LH. Most of the time, the target cells will only receive enough signal to be stimulatory. And so what happens in those cases, when a target cell receives a stimulatory dose of hormone like LH, for example, they also will desensitize over time. And this will be a lot faster, okay, and doesn't take a lot of hormone. It's still with low concentration. So important concept here is desensitization or internalization of receptors can happen with very high levels. That tends to be slower, and with lower levels it tends to be pretty rapid. Okay, and the first thing that can happen is there's a decreased affinity for the hormone by the receptor. Okay, and you can have a decreased affinity of the hormone receptor complex for G proteins. So first of all, the hormone receptor can be internalized. There can be less affinity for the hormone receptor, for the hormone by the receptor, or there can be less affinity of the hormone receptor complex for G proteins. So G proteins could fall off. Well, these are independent of cyclic AMP. And so for a receptor to be receptive for a hormone, it first must be phosphorylated. So the point here is that LH could work through multiple second messenger systems. Its favorite is cyclic AMP, but it could work through others to maybe turn off stimulation of the target tissue. So once we got the target tissue stimulated, maybe further binding of LH to the target cell would actually turn off this signal. And so that's something that's pretty deep and heavy into endocrinology, but just to let you know that target cells won't respond to FSH or LH continuously. Okay? Finally, we'll talk about basal levels. Okay? So basal levels of hormone, we're always going to have a little bit of hormone in the system. Basal levels of hormone muffle the response to acute increases of similar levels. So let's say we had a half a nanogram of LH running through the system. Well, if it spiked up another half a nanogram, it's not going to be a stimulatory dose. So the basal levels kind of hang out around just to muffle any acute increases of similar magnitude. And this seems to be independent of receptor number and independent of cyclic AMP. So there's a check valve on the system, if you will. There's a control level on the system, and what drives that control level to keep a target cell from responding when it shouldn't be responding to the stimulatory dose, it's these basal levels. So why are hormones released in low levels continuously? It's to just kind of keep the system running and muffle any response to very short-term increases of similar levels. So there's 3 points on this slide. First, very high levels obviously will downregulate the target cells, and this tends to be a slow process. However, stimulatory doses also desensitize or downregulate the response, and this tends to occur a little bit faster and at much lower concentrations. And usually what happens is the hormone receptor complex and G protein affinity for each other decrease, and so there's no method to carry on the system. If you don't have G protein attached to this receptor, it's not going to respond. Kind of like we talked about with GnRH. Okay? And then you got basal levels. So you got very high doses, which are experimental in nature. You get stimulatory doses, which are natural in nature. Okay? So like during an L8 surge, those target cells will become desensitized pretty quickly over time, because desensitization also occurs at stimulatory doses. So this level here would be even more higher than an L8 surge. Okay, this might be 2 L8 surges. So we got very high, we got the normal stimulatory doses, and then we got the basal levels, which kind of keep things in check and don't let little increases in hormone cause a main effect. So we need a stimulatory dose, not just a doubling of the basal levels. So I hope that makes some sense to you. Okay? All right, let's talk about some structure-function relationships. So for the gonadotropins, okay, their half-life is only about 20 to 220 minutes, so less than 30 minutes to up to 2 hours in the serum, in the circulation. That's for LH and FSH. It tends to be longer for HCG and ECG. That's why we can combine these two into a product called PG600. So if you want to Google that, PG600 is a hormone we give to postpartum sows and prepubertal gilts to stimulate follicular growth and ovulation. So ECG or PMSG will bind to FSH receptors at high doses, and HCG will bind to LH receptors at high doses. So the half-life for these two are longer because they've got more carbohydrate attached. And when we look at just these two, HCG has a longer half-life than ECG or PMSG. So what happens is we give one shot to a pig. The first thing that happens is ECG binds to FSH receptors and stimulates follicular growth. And then it's degraded by the body and downregulation occurs. Well, what's left in the system is HCG, which will bind to LH receptors and cause those gals to ovulate. Okay? And that's what I mean down here. So hence both ECG or PMSG and HCG work extremely well for superovulation or just stimulation of follicular growth and ovulation in domestic animals or laboratory animals. Okay? So we use this a lot, except for beef cattle who are used in embryo transfer. In those cases, we don't tend to use ECG or PMSG. We really use porcine FSH, which is harvested from pituitaries from the pig slaughterhouse. So you've been reading about these slaughterhouses that have been closing down. That slaughterhouse in Sioux City, Iowa, kills 20,000 pigs a day. So a biomedical company could go to that slaughterhouse and collect 20,000 pituitaries every day, and they could take those pituitaries back to the lab, mince the tissue up, grind it, purify it, and get all the FSH out of those pig pituitaries, and that can be used to superovulate cattle. But my point here is that because these have more carbohydrate attached to them, because they're glycoproteins as well, because they do have more carbohydrates attached to them, because they have longer half-life, hours to days, they work really well for superovulation or synchronization of, say, postpartum sows and prepubertal gilts, and that product's called PG600, and it contains both of these. So here's the cool part. If we remove some of that polysaccharide side chain of HCG or PMSG or ECG, they become inactivated. They can no longer bind to the receptors. Okay? So what happens to those hormones if we modify or remove parts of the sugar side chains is these hormones would be captured, internalized, and degraded by the liver or removed by the kidney due to the small size. Okay? For example, the early pregnancy test kits that women buy at the drugstore or grocery store to determine if they're pregnant or not, just a little bit of side note here. These early response kits, those kits test for this hormone right here. Because about day 18 of pregnancy, the chorion will start to be formed. That's what the C stands for. And once the chorion's formed, it'll start producing this glycoprotein. In fact, it's really a gonadotropin, which is also a glycoprotein, called HCG. So that's just some side information. So this is a very important hormone for a lot of reasons, for early pregnancy detection in humans, and it works well because at high doses it'll bind to the LH receptor and it will cause ovulation. Okay, let's go back to the subunits. Remember, we got an alpha subunit, we got a beta subunit. Okay, so it's the protein part, it's the alphas and the betas that are important for receptor binding of these gonadotropins. Okay, in fact, binding is even better and occurs easier if the sugars are removed, because the sugars create a little electrostatic repulsion of the hormone from the receptor. But if you take the sugars off of LH or the sugars off of FSH, they will not affect the target cells. Okay? The beta subunit determines the biological specificity and activity of the hormone. So remember, the beta subunits are the ones that are unique in these hormones. Alpha subunits are kind of generic, beta subunits are unique. And so The beta subunit determines the biological activity of the hormone. So whatever the beta is, that's the hormone. So if we got an alpha and a beta LH, it's LH. If we got an alpha subunit and a beta FSH, it's going to be FSH. But we could separate those two molecules and switch them, and they would still work fine with each other's alpha subunit. Okay? So because we have two different proteins hooked together as a functional hormone, it's called a heterodimer. Hetero meaning different, like heterosexual, and dimer meaning two proteins. So it's a heterodimer. So all these gonadotropins are heterodimers. All of them have an alpha and a beta. That includes ECG and HCG, LH and FSH. Okay. So when they're separated into separate alpha and beta subunits, they look different as compared to when they bind together. So when an alpha binds to a beta, a dramatic conformational change occurs in both subunits, which makes them unique and makes them fit nicely into that receptor that I showed you at the first part of the lecture. Okay, so next we're going to look at figure 7, and that's the last figure in this chapter. So there's been a couple of theories, actually 3 theories that have been proposed about these gonadotropins and their receptors. Okay. The first theory was that the beta subunit binds the receptors, but only with the alpha subunit attached. So that was the first theory. So they said, okay, the beta subunit binds to the receptor, but it won't bind to the receptor without an alpha attached. And then the second theory was that both alphas and beta subunits, both of them bind to the receptor, but the beta subunit carries the specifics for that hormone. And the third theory, which was developed looking at comparative studies with PMSG or ECG and LH, all from the horse. So they were comparing PMSG, which is obviously from a horse, and ECG for equine ECG, and that's led to the negative specificity theory. So that's what's shown in figure 7. So that's probably the one I would lean towards. But it's important because it could be all 3 theories, and it could be just one of the theories. So just to let you know, there's 3 theories. One is beta binds to the receptor, but only with the alpha attached. I don't tend to believe that one. The second theory, which is kind of in between, it says both alpha and beta bind to the receptor, but B still carries the specifics. And then there's the third theory, which is called the negative specificity theory. So if I ask you about the negative specificity theory, you'll know that's what we see in figure 7. So what the negative specificity theory states is that it's the alpha subunit that binds to the receptor, but the specificity resides within the beta subunit. So it's kind of similar to the second theory. And if you believe this, then you got to say, okay, the common alpha subunit that's in LH, FSH, and even TSH, and in ECG and in HCG, that alpha subunit has got to have extremely high affinity with all glycoprotein receptors. But it does not discriminate. And that helps explain why high doses of FSH will actually bind to LH receptors. High doses of LH will actually bind to FSH receptors, and high doses of ECG will bind to FSH receptors, and high doses of HCG will bind to LH receptors, and that's why it works in those pigs. Okay? So there's no discrimination by the alpha subunit. The alpha subunit simply loves those 7 transmembrane domain receptors, and that will bind. Okay? So the alpha subunit has high affinity. In theory 3, the alpha subunit has high affinity with all glycoprotein hormone receptors. Kind of on the flip side of that, still in the third theory, is that the beta subunits only have marginal high affinity interaction with the receptor, but it's the beta subunit that ensures the strict specificity by prevention of the hormone binding to the wrong receptor. So that's because of beta LH doesn't fit nicely in an FSH receptor. That's what keeps LH out of an FSH receptor. So that's kind of important. So again, the three theories. One, it's the beta subunit that binds, but it'll only bind with an alpha attached. Two, both bind, but beta carries the specifics. And then what's shown in figure 7 is the negative specificity theory, where that theory states that the alpha subunit will bind to any glycoprotein hormone receptor. It's the beta that has the specific. So I think the next slide should be figure. Yep, there it is. Okay. So you may have to pull yours out and put on your glasses in a magnifying glass. Okay? But we'll start. And this is a cartoon, this is a cartoon, this is a cartoon. So don't think, we know what the receptor looks like, but here's a cartoon. Okay, so this is a negative specificity theory or model of the interaction of glycoprotein hormones with their receptors. And so here we have an LH receptor. Notice that it has a wall up here. Okay, here's the FSH receptor. Notice it has a wall up here. And what you got to do is believe that these hormones won't flip, not 180 degrees. So here's alpha LH and here's beta LH. So here's LH. Here's alpha FSH. Here's beta FSH, and so they're bound together. Remember, they're non-covalently linked. And all this is trying to show us here is that there is alpha and there is alpha, and these two alphas are identical, and notice they fit nicely in this little notch here in this cartoon. So there's no doubt that if these two proteins have affinity for each other, now not shown here are the sugar side chains, but if you can see where alpha fits nicely in there, you see where alpha nicely fits there, but same token, this alpha would definitely fit nicely in there, and this alpha would fit nicely in there. Okay? So it's the beta subunit that has this little thumb sticking off of here, and you can see that LH, as drawn in this picture, would only fit with the LH receptor because this knob here would come in contact with that inhibitory site. So this hormone, if this hormone tried to fit in there, these two would oppose each other and not fit together nicely. Whereas this bump or little side node here is sticking out on the right side, and it can sit right down there nicely, activate the LH receptor, and off we go. Okay, so here is the LH receptor. It's bound to LH, and it would take a lot of FSH to get over here and cross over, which can happen, but it has to be very high doses. And then you can see here LH would not fit in this receptor, just like FSH wouldn't fit in this receptor, because this bump on the side would conflict with that receptor specific inhibitory site. So that's going to avoid that. That's going to love to bind here. That's going to avoid that, and that's going to love to bind here. So here we have FSH, alpha, and beta bound its receptor. So let's just read this paragraph. Numerous experiments have shown that the two subunits interact with the receptors, but the alpha subunit seems to be principally involved. The alpha subunit seems to be principally involved with that extracellular domain. Okay? Indeed, the alpha subunit appears to be sandwiched between the receptor and the beta subunit in the hormone receptor complex, since it's no longer accessible to anti-alpha monoclonal antibodies after binding of the hormone to the receptors. What they're saying is, if you raise antibodies against the alpha subunit, say this is in culture or even in a live animal, and you flood this area with antibodies against that alpha subunit, those antibodies can't bind to that, and so they won't stay stuck here. So, if you're tracking where this sucker went, like if you flooded this area with antibodies, those antibodies would come over here and bind. Okay? But once the hormone's bound its receptor, the alpha subunit is protected and well hidden by the receptor as well as the beta subunit, and no antibodies can attach, and that's what they're saying here. Okay? So Okay, so it was no longer accessible to anti-alpha monoclonal antibodies after binding of the hormone to the receptor. Besides, most covalent bonds promoted by the bifunctional or photoactivable reagents between the hormone and the receptors involve the alpha subunit. Okay, so they've pretty much shown that it's the alpha subunit that binds to the receptor most tightly and with most affinity. So therefore, the interaction of the beta subunit with the alpha subunit has two distinct complementary functions. Okay, so why does alpha and beta have to bind? Well, once they bind, alpha and beta, it induces the active conformation of the alpha subunit, allowing high affinity binding to the receptor. So once alpha and beta bind together, it makes alpha look like something like that, which has a lot of affinity and fits nicely into the receptor. Number two, why do we need alpha and beta bound together? Well, the second reason is beta discriminates the receptor to which binding is authorized by inhibiting the interaction to other receptors. Okay? So that's why if we stuck a beta LH onto an alpha FSH, it would still bind to an LH receptor. And that's why if we stuck a beta FSH onto an alpha LH, it would still bind to the FSH receptor. So that's what I mean by specificity. So you can think of this block diagram here. We got to have the bump over on this side if we're FSH, because this will block the bump, and we've got to have the bump on this side if we're talking about LH, so it fits nicely in here. So I hope that's not too confusing, but that's the third theory, and that's probably the most accurate. Okay, the negative specificity theory is that alpha and beta need to be bound together. When they leave the pituitary, they need to circulate in the blood, and once they get to their target tissue in the gonads, there's going to be two receptors there, and by and large, LH binds to the LH receptor because of that negative specificity, and FSH binds to the FSH receptor because of that negative specificity. Okay? Okay? So here's the inhibitory site, here's the inhibitory site. Inhibitory site, inhibitory site. So it'd be really challenging for FSH to bind to LH receptor and very challenging for LH to bind to an FSH receptor. But it will occur at high doses. I'm just telling you that. At really, really extremely high doses. You can force that into there. Okay, so one more time, and then we'll take a little break. One more time. Interactions of the beta subunit with the alpha subunit has two functions. You got to have them together, bound together, to induce the active conformation of alpha, allowing for high affinity binding to receptors. And number two, it's the beta subunit that has the specificity and discriminates the receptor to which binding is quote-unquote authorized by inhibiting or somewhat inhibiting the interaction with other receptors. Okay? And the alpha subunit must be sandwiched between the beta subunit and the receptor because of if you raise anti-alpha antibodies, they will not bind after that alpha-beta heterodimer is bound to the receptor. Okay, so we're going to take a little break and I'll be right back. All right, we're back. Okay. So, you may be asking the question now, well, if there's so much specificity between native LH and native FSH in a particular animal, like a cow or a sheep or a goat or a pig or a human, whatever, then why on earth does ECG and HCG and even equine LH have the ability to bind to one or both receptors? So why can ECG, HCG, and equine LH bind to either the LH or FSH receptor in other species, and that allows us to use them for superovulation or induction of follicular growth and ovulation? Well, they must have, ECG, HCG, and equine LH must have different positions of that thumb sticking out there, which is inhibitory site, that are ineffective in non-equine or non-human species such as the rat, pig, sheep, and cattle. So what we're saying here is because of the different inhibitory position sites, either on the hormone itself or on the receptor, probably more importantly on the receptor, those positions are somewhat different and don't block these three hormones from binding to either one of these receptors. And so these three hormones have been used in the rat, the pig, the sheep, and cattle. And they, remember, E's come from equine and H comes from human. And so there are different inhibitory sites in the receptor, that crosshatched area we saw in figure 7 in the rat, pig, sheep, and cattle, that don't prevent these three hormones from binding to these two receptors. Okay? All right, so what are the sugars for? What are the sugars for? Why are they glycoproteins, and what are those oligosaccharide chains hanging off these two hormones? Well, I've already told you that those sugars are not needed for binding to the receptor. In fact, the hormone binds better without them. But the sugars are needed for membrane transduction of the hormone signal. So what that means is that sugars may turn on G protein or help with adenylate cyclase activation in a cyclic AMP mediated second messenger system. And most importantly, the sugars that are needed for membrane transduction of the signal, they're the sugars that are different from those that are responsible for the longer half-life. So in general, the sugars attached to a glycoprotein protect that protein from enzymatic degradation and extend the half-life. But there's also sugars on there that are needed for membrane signal transduction. So they carry on the signal, and probably it involves G protein or adenylate cyclase activation. Why do we know this? Why do we know this? Well, if you totally take off the sugars off of LH, HCG, or ECG, that's the deglycosylation. So if you deglycosylated them, took all the sugars off, they still bind to the receptor, but they don't have any membrane transduction of the hormone signal, so they would sort of act like a what? Antagonist. In other words, occupying a receptor, but don't tell the target cell what to do. Okay, so deglycosylated LH, HCG, and ECG, when you give them, you're not going to, for example, let's say you deglycosylated LH and gave it to a cow that's ready to ovulate. She wouldn't ovulate. It'd be just the same as giving an antagonist. So you would occupy the receptor, you would block that receptor, you would block normal glycosylated LH from binding, and nothing would happen. Okay? So the sugar structure and the sugar locations are also important. Okay? Let me tell you. For example, let's go back to the horse. We got equine LH and equine chorionic gonadotropin, or PMSG. They have the same beta subunit and the same alpha subunits. So this will blow your mind. Equine LH and equine PMSG, or equine ECG, or equine chorionic gonadotropin, they have the same beta subunits, they got the same alpha subunits, but they differ in potency in stimulation of rat Leydig cells in culture. So Leydig cells are the cells in a testicle that make testosterone, and they have LH receptors. Okay? So if you get some rat Leydig cells and make a culture of rat Leydig cells and give one petri dish equine LH and give the other petri dish equine chorionic gonadotropin, they're not going to make the same amount of testosterone, even though they have the same beta subunit and the same alpha subunit. Well, how is equine LH and equine chorionic gonadotropin, PMSG, how are they different? They have they're structurally different due to the carbohydrate chains. Okay? So another example of not only is the alpha subunit important, not only is the beta subunit important, but the sugars that are attached to each are also important. And there are specific sugars on these glycoproteins that are needed for what? Membrane transduction of the hormone signal, getting that signal into the inside of that cell. The other sugars that are out there are simply for protection and extension of half-life. Do any alphas here require sugars to bind to the receptor? Heavens no. Nope, they do not. In fact, they bind better without. Okay? So here we got rat Leydig cells in culture. We gave one dish equine LH. We gave the other dish equine chorionic gonadotropin, which have identical beta and alpha subunits. Protein-wise, amino acids-wise, but they differed in carbohydrate change. And so I don't know which one does better, but one of these is better at causing Leydig cells to secrete testosterone than the other. Even though they're the same alpha and beta subunits, they just have different sugars attached. Okie doke. Here's another example. Human. Okay? Human HCG and human LH exhibit a binding affinity for rat Leydig cells equal to equine chorionic gonadotropin. So human chorionic gonadotropin and human LH will bind the same as equine chorionic gonadotropin for rat Leydig cells. But the estimated dose, the dose of HCG and human LH, the estimated dose to stimulate a 50% increase in cyclic AMP or a 50% increase in testosterone secretion. So the dose needed for both of these is 60 to 100-fold less than that of ECG. Why? Because of the different locations and amounts of sugars attached to these two hormones. Okay? So we can even see superactivity. So here's an example of superactivity. Equine FSH can stimulate rat Sertoli cells, which are another cells in the testicle of a rat and all male mammals. And Sertoli cells babysit sperm and help with sperm production. Okay? So if we got rat Sertoli cells in culture, and we give equine FSH and other species FSH, equine FSH can stimulate rat Sertoli cells in culture better than any other FSH. Why? Because of their sugar side chains are more efficient at stimulating that transmembrane signal to activate what goes on inside the cell. So all I'm showing you is all this stuff is the sugar part of these glycoproteins is extremely important. So the answer to both questions is the same. Okay? First of all, these are binding FSH receptors and these are binding LH receptors. Okay, so these two are binding LH receptors in Leydig cells. Equine FSH is binding to FSH receptors in Sertoli cells. So we already established that the inhibitory structure is such that these hormones can bind to other species without any interference. And now I'm showing you that these two hormones and this hormone can affect cells differently, and it all has to be because of the oligosaccharide, the sugar side chains. The carbohydrate part of these hormones is doing a better job of stimulating that transmembrane signal. The last topic I want to talk about in chapter 3 is isoforms. So let's first define what an isoform is. So there are gonadotropin isoforms. A protein isoform is any of several different forms of the same protein. So an isoform is a protein that's similar to another protein, but has somewhat of a different amino acid sequence, but it's similar enough that they look very much alike and sometimes will act alike. So let's continue. Different forms of a protein may be produced from very closely related gene duplicates. So we know we have a gene for LH, we know we have a gene for FSH. Well, there might be a gene duplicate that's very close to LH and very close to FSH, down the line of a chromosome. And these very similar genes can cause the transcription and translation of an isoform of LH or an isoform of FSH, for example. So different forms of a protein may be produced from a very closely related gene duplicate as same protein or a protein makes no sense for highly diverged homologous sequence that arose from a single ancestral gene that duplicated billions of years ago and subsequently diverged greatly in sequence, structure, and functionality, or may arise from the same gene by alternate splicing, so mixing up the exons. Okay, so let's read that again. So it really doesn't make sense to have isoforms, is what it's saying. Okay? So why make the same protein again, or why even make a similar protein? It makes no sense for a highly diverged homologous sequence. Okay? So these duplicates arose from a single ancestral gene that duplicated billions of years ago in time and subsequently diverged greatly in sequence, structure, and functionality, or may arise from the same gene by alternate splicing. So there's two ways to get an isoform. You could have a gene duplicate somewhere else in the DNA, or you could somehow change up the putting together of the exons. So instead of going from 1 to 11, let's say in that hormone receptor, that glycoprotein hormone receptor where we had 11 exons, maybe you switched 10 and 11, or maybe you switched 9 and 8 exon. That's what an isoform is. Okay? So you have to be aware that there exists isoforms of protein hormones. There's no such thing as isoforms of steroid hormones, but there is such a thing as isoforms of protein hormones like LH and FSH. And so these glycoprotein isoforms or gonadotropin isoforms may be secreted by the anterior pituitary gland, and they may differ according to the reproductive state of the animal, and thus make another control point in regulation of gonadal activities. So in other words, the pituitary during the LH surge could be putting out a lot of LH isoform that could kind of dampen the effect of the LH surge, for example. So what are these isoforms doing, and what is their purpose? They do exist. Okay? And where do they come from? They come from the anterior pituitary gland, and these isoforms are going to differ according to the reproductive state of the animal. And so these isoforms, these gonadotropin isoforms, could provide another control point to regulate gonadal activities. Most likely, these isoforms can affect the stability of the stimulatory state of the hormone receptor complex. So, some isoforms could be like an agonist, whereas other isoforms could act like an antagonist. So, some isoforms could, maybe superstimulate the target tissue as target cell as compared to the native hormone LH, or some may dampen the signal. Okay? So, what would be the purpose of a subtly different alpha subunit or a subtly different beta subunit in a gonadotropin isoform? We can't rule out the fact that the purpose of the protein portion of the gonadotropin isoform may participate in membrane signal transduction, kind of like we've been talking about the sugars. And these isoforms may also be in charge of swapping out of the disulfide bridges with the receptor, or the different isoforms having a different structure could have a different location, could lead to a different three-dimensional location within each, and could change the conformation and help with signal transduction. So isoforms of the gonadotropins can maybe help or maybe hinder the normal transduction of the message that LH and FSH would have for the gonad. But I just want you to be aware that isoforms are out there, they exist, and the reproductive system is not immune from isoforms because we do have proven gonadotropin isoforms, and we know they come from the anterior pituitary gland. So their isoforms are just additional proteins that look and somewhat can act like the native hormone. And so they're out there and they exist. Okay? So when you're looking at these big glycoprotein hormones like LH and FSH, the protein sequence, the amino acid sequence of alpha and beta are important. How the beta and alpha interact with each other are important. The location and purpose of the sugars is important. For the most part, the sugars are for membrane transduction of the signal. And who knows, maybe the gonadotrope isoforms are better at membrane signal transduction than the native, or they could be worse. And so they exist out there. They are secreted. They are flowing through the blood system. And they can differ according to if she's maybe getting ready to ovulate or she's got a CO present. And all I'm saying here is gonadotropin isoforms secreted by the pituitary gland provide another control point in the regulation of the ovary and regulation of the testicle, and they exist. Okay? So that's enough for this chapter.